Good morning, everyone. Sorry about the little mix-up this morning. Greg, thank you for always having extra material. So today I'm going to be talking about studying apoptosis through the CyTOF, and I'll talk about my specific project, and hopefully you can ask a lot of questions after if you're interested with some of the more nitty-gritty of the techniques I use in order to get these results. So the focus of my talk is going to be about a molecule called TRAIL. And TRAIL is a TNF family member that functions as a ligand to induce apoptosis. You can basically think of TRAIL as your body's own targeted cancer therapy. And just to give you a little bit of background about this process and what I'll be talking about, TRAIL signals through a canonical apoptosis-inducing mechanism. Actually, is there a laser pointer? I can just use my hand.
No, I was just using the pointer.
Okay. There's an actual pointer?
There's a wooden one.
Oh, okay. Yeah, no, it doesn't matter. It's okay. So basically, TRAIL oligomerizes here, and it forms this thing called the DISC, the death-inducing signaling complex, that homodimerizes cleave caspase 8, and then leads to this thing BID become truncated to form TBID, inhibits BCL2, forms to these two things to come together on the surface of the mitochondria, and then this leads to cytochrome C getting extruded from the mitochondria, and then leading to cleavage of all of these caspases, and eventually the cell implodes in apoptosis. And all of this is masterfully orchestrated and has been studied extremely well over the last 20 years. This is a very well-defined process. But what's not really well-defined is all these upstream signaling kinases and proteases that are involved in the process. We know that they're implicated, but we don't really understand how. And part of the reason that we don't understand how all these other upstream proteins regulate TRAIL-induced apoptosis is that TRAIL-induced apoptosis happens asynchronously, even in cell lines. This is a paper from Nature from a few years ago, and each one of these bars represents a cell that was given TRAIL right here. And this shows how long the cells take to die. And the first cells start to die about 140 minutes, and the last cells die at about 300 plus minutes. So there's a huge amount of variation. And so what we were able to do by the CyTOF is basically take the fact that these cells are asynchronous and understand it using single-cell methods by aligning them. And in order to do that, you need to have enough markers that you can record some of the markers by which the cells are dying along these canonical apoptosis cascade, and still also have other parameter space for looking at the other markers of interest. So if you think of at any time point, because the cells are dying in this asynchronous way, there's going to be cells at different stages of the process. So even right after you give TRAIL, there's going to be some early cells kind of indicated by those light blue ones that are going to be already starting to die. And in the kind of middle time points, there's going to be one cell like right here that's already almost dead, and a lot of cells that look like they're kind of resisting the process. And this is kind of the Western blot problem. When you do these things by Western blot, all of these cells at different points of the timeline are still pooled together. So what we did is we pooled the data, we just combined it all, and then we re-extracted it based on these canonical apoptotic signaling markers. And then reorganized it based on the true progression of apoptosis, as opposed to based on just where these things were dying in chronological time. Then from that, we created network maps, and these maps basically just show the correlation of all of the markers we measured by CyTOF with all the other markers. And by looking at the correlations, you can see how things in the network relate to each other and try to understand the causative effects. The other thing is, once these cells are in the process, we can look not only at the process of apoptosis, but also the cells that are stalled in the apoptotic cascade. And this kind of gets us to the dirty little secret of this previous work on TRAIL-induced apoptosis. So remember that Nature paper that I showed you before with the bar graphs? This is supplementary figure 4 from it. And you see that all these cells die in this kind of nice unimodal Gaussian curve, like that other graph suggested. Now, part of the issue here is that this is achieved with the addition of cyclohexamide, which inhibits translation and transcription and also induces DNA damage. In addition, these cells are actually also serum starved, so it's not exactly the most physiological set of conditions. But by treating these cells in this way, you're inducing a host of secondary effects, as you would imagine, by adding cyclohexamide. And B, if you do this, you're missing a lot of what I would consider the really interesting part of trail-induced apoptosis. Because if you don't add cyclohexamide and you treat them under normal conditions without serum starvation, many of the cells refuse to die. Actually, 10% of the cells. And if you're thinking about this as your body's own chemotherapy and about it as a chemotherapy that's used in the clinic, and this has been used in dozens of clinical trials, 10% of your tumor surviving is a whole lot of tumor. So I decided to focus not on these cells that were focused on before, the ones that were dying, but use the methods that we developed at the CyTOF to focus on these cells that they don't show you, the cells over here that never respond to trail. So we've looked at different ways to subset these cells, and a really interesting thing is that there's this whole population that doesn't die, these apoptotic resistors that are cleave PARP negative and cleave caspase-3 negative, and they correlate almost perfectly with the cells that have really high phospho-S6. And it's not just that these cells aren't dying, the S6 isn't going down, you actually see the S6 getting induced in these HeLa cells over 12 hours. And so, we wondered, what happens if you look at the kind of map that I showed you before, where we look at the relationships between S6, which seems to be central to this process, and also all these other survival factors, mostly MAP kinases, and also their relationship to the downstream suite of proteins responsible for executing apoptosis. And I think the cool thing that the CyTOF enabled with this research is everything in an orange box is something we measured simultaneously in single cells. And there's no other approach available where you can look at all these things on the protein level at one time. And there would be no other way to understand this network because of the asynchronicity without doing it all simultaneously in single cells. So everything in orange is something we measured. Everything in purple, actually, we now have antibodies for, but we didn't as of a few weeks ago. So these are just nodes between nodes we measured, where we basically looked at the a priori knowledge and said, okay, we know that phospho-AMPK doesn't phosphorylate S6, but we know that phospho-AMPK phosphorylates S6 kinase, and as you can tell from the name, we know S6 kinase phosphorylates S6. We just put those in the middle to take in all the a priori knowledge that exists. And the red lines are negative correlations, and the green lines represent positive correlations. So we created this map, and we wanted to see if we could use it to generate testable predictions. And since I just said, since S6 phosphorylation is strongly correlated with survival, we thought that perhaps if we could inhibit S6 with a PI3 kinase inhibitor, which lies upstream of S6 on that map, then maybe we could also remove the apoptotic resistant population. And I was super excited when I first saw this pair of histograms. I feel like, I don't know if you guys have worked with inhibitors, they rarely work this nicely. But this works brilliantly. None of these cells have induced S6 in this population. There's total ablation of that population. And then I looked at the survival data excitedly, and I thought that in the same way, all the survivors would be taken away. And of course, that's not at all what happened. Because this is a viability marker. This is cisplatin. And all of the cells that are high in cisplatin are dead. But all the cells from here over are still alive. And there seems to be, in these histograms, no significant change between the cells that have high S6 and the cells that don't. Both populations show a large level of survival. And so we went back to the network map to try to understand what was going on. And kind of after I'd gotten over the fact that my cells didn't die when I wanted them to, and I looked back at this map, I kind of realized this makes sense because it's not just PI3 kinase that lies upstream of the pro-survival network. There's also a whole other level, a whole other number of MAP kinases that are inputting signals to this network. And so what I want to do is I want to just show you the CyTOF data, but everything that is static between the correlations between these proteins, I'll just gray out to make it a little visually easier to digest. So everything I just grayed out is the same between all of my conditions. And so this is what happens what the survivors look like when you add trail. RISC inhibits phospho-BAD, phospho-AMPK inhibits S6 kinase. RISC2 probably is phosphorylated p53. It goes in that direction. Now, when you add the PI3 kinase inhibitor, there's a radical compensatory change in the network. Instead of RISC1 inhibiting the phosphorylation of BAD, it looks like it actually has an excitatory relationship, and it also looks like there's a new connection here with phospho-BCL2. In addition, there's a new connection between S6 kinase and phospho-AMPK, and through an unknown mechanism, cleave caspase 8 and phospho-S6 suddenly have a positive correlation. And although the vast majority of these factors stay the same, the relationship between the survival factors and the apoptotic regulators shifts so that although PI3 kinase is being inhibited, other factors appear to compensate in order to maintain survival in this resistant population. So, yeah, the takeaway is that the network seems to be very robust. So that although one kinase is inhibited, others seem to be able to take up the slack and maintain survival. And so we were wondering what happens if we shut down multiple upstream signaling nodes. Can we strategically target these other pro-survival target kinases in order to ablate survival? And so what I want to show you now is a clonogenic assay. This is the most low-tech thing in the world. You just plate your cells out at a really low density, and then you add whatever you want and see how many survive. And it's a very simple but really sensitive way to look at survival. And so this is with no treatment. This is with trail. You see about 10% of the cells survive, about 200 colonies. This is with trail plus cyclohexamide. This is what was in that Nature paper. Kind of a noticeable difference between just trail alone. And this is trail plus this P38 inhibitor, which is another one of the inhibitors we used. And actually, any of the single inhibitors that we employed, PI3 kinase, P38, MEK inhibitors, actually show pretty modest changes over the trail alone. Trail, just when you add trail alone. So this is trail plus this inhibitor. But now when you add 2 inhibitors and then 3 inhibitors, you're able to totally ablate survival. And this is just a bar graph showing you that. So now what I'm doing is I've developed the other antibodies in that network that were not measured previously. And I'm going after and seeing how does the network change when you do each of these combinations. And what are the most effective strategies in order to knock out all of the minimal number of kinases in order to ablate survival of the system. So thank you for listening. Sorry for being late again. Big thanks to Greg Bebahani, who filled time for me before, and whose work on the cell cycle really helped inform this. The bioinformatics was done by a professor now at ETH Zurich named Manfred Clayson. And big thanks to Gary and the rest of the Nolan lab. So I'll be taking any questions. Thanks. Yeah, Ron.
So just back to where you had the timeline.
Yeah.
Different timers. So did you collect each of those at separate points and then just combine the files so you could extract out?
Yeah, exactly. I kind of glossed over that, but basically what I did was take like a dozen time points and then just pool all those cells together. I mean, I could have just run them all at one time, but just informatically we took those data files and then pooled them. And then using the markers we used were NF-kappa B to capture the early events. We have apoptosis and cleave caspase 3. We basically use those kind of as signposts along the way, and align the cells based on the presence of those markers. Cool. Great, I'll be around after.